Hello, and welcome to your Microbiology Bio 203 video lecture on the functional anatomy of prokaryotic and eukaryotic cells. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. To begin, we need to define the terms prokaryote and eukaryote. The term prokaryote comes from the Greek word for pre-nucleus. Organisms labeled as prokaryotes lack a nucleus and any membrane-bound organelle. The term eukaryote comes from the Greek word for true nucleus. Organisms labeled as eukaryotes have a nucleus and membrane-bound organelles. This is a complete list of characteristics of both prokaryotes and eukaryotes. Prokaryotes have one circular chromosome not in a membrane, no histones, no organelles. The bacteria use peptidoglycan for their cell walls, the archaea pseudomerin. They divide by binary fission, which is very similar to mitosis, but because they lack a nucleus, it is described in a different fashion. Eukaryotes have paired chromosomes in a nuclear membrane. Their DNA is associated with histones. They have membrane-bound organelles, polysaccharide cell walls when present, and they divide by mitosis. The prokaryotic cell. The prokaryotic cell averages 0.2 to 2.0 micrometers in diameter and 2 to 8 micrometers in length. Most bacteria, or prokaryotic cells, are monomorphic, which means they come in a single shape. Few are pleomorphic. Some examples of bacterial or prokaryotic cells and their shape include the bacillus, coccus, spiral, star-shaped, and rectangular. Bacterial cells, or prokaryotes, may also form interesting cellular arrangements where the cells are paired, clustered, in chains, groups of 4, or cube-like in groups of 8. If they are paired, they are referred to as diplo. They might be diplococcus or diplobacillus, depending upon the cell shape. Clusters are staphylo, chains are strepto, groups of 4 are tetrads, and cube-like groups of 8 are sarcinae. Many prokaryotes are named based on their shape. The scientific name bacillus is referring to the shape of the cells which carry that name. External to the cell wall. In prokaryotes, we have a glycocalyx. This is a structure external to the cell wall, which is often viscous and gelatinous. It can be made of polysaccharides or polypeptides. There are two types of glycocalyx, one which is neatly organized and the other which is not. The neatly organized glycocalyx is referred to as a capsule. The unorganized is referred to as a slime layer. The glycocalyx may also contribute to the virulence of the organism that possesses it. Capsules can prevent phagocytosis, and extracellular polymeric substances can help the organisms form biofilms, which also help resist phagocytosis. Flagella. If a prokaryote is to be motile, they will possess a flagellum, and only a flagellum. Prokaryotes do not grow cilia. A flagellum is a filamentous appendage external to the cell and can propel the bacteria. It is made of the protein flagellin. Flagella have three parts: the filament, which is the outermost or hair-like region, a hook, which is attaching the filament to the cell, and a basal body, which consists of the rod and pairs of rings and anchors the flagellum to the cell wall and membrane. This is an illustration of the different types of filaments, or flagellum, that prokaryotes can possess. Flagella allow bacteria to move toward or away from a stimulus in what we refer to as taxis. Taxis can be related to chemicals or light, at which point we add a prefix such as chemo, and the term becomes chemotaxis. When flagella rotate to run or tumble, the microorganism is said to be moving in a direction with purpose. This is an illustration of a bacteria running and tumbling. Axial filaments are also called endoflagella and are typically found in spirochetes. They are anchored at one end of the cell, and the rotation causes the cell to move like a corkscrew. This is an illustration of an axial filament in a spirochete. Fimbriae and Pili. Fimbriae are hair-like appendages that allow for attachment. Structurally, they may appear similar to a flagellum. However, they are not used for locomotion, only attachment. This is an illustration of Fimbriae. Pili are involved in motility, gliding, and twitching motility, and in conjugation, which is a form of sexual reproduction in prokaryotic organisms. Conjugation pili allow for the transfer of DNA from one cell to the other. The cell wall. In prokaryotes, the cell wall prevents osmotic lysis and protects the cell membrane. In prokaryotes, it is made of peptidoglycan, and it can also contribute to pathogenicity. Peptidoglycan is a molecule or polymer of repeating disaccharide rows with N-acetylglucosamine and N-acetylmuramic acid at its base. The rows are linked by polypeptides in a fashion illustrated here. The cell wall can be used to distinguish prokaryotes into two groups, the gram-positive and gram-negative group. The gram-positive organisms will have a thick peptidoglycan and layer and teichoic acids to hold it together. The gram-negative group will have thin peptidoglycan buried under an outer membrane with a periplasmic space. Teichoic acids. Teichoic acids are lipoteichoic acid links that hold the cell wall to the plasma membrane, while teichoic acids link the peptidoglycan to each other, and teichoic acids carry a negative charge to regulate movement of cations. Polysaccharides and teichoic acids provide antigenic specificity in prokaryotes. This is an illustration of a gram-positive cell wall. Gram-negative cell walls have a periplasm between the outer membrane and the plasma membrane, which contains peptidoglycan. The outer membrane is made up of polysaccharides, lipoproteins, and phospholipids. Gram-negative cell walls protect the cells from phagocytosis, complement, and antibiotics. They can be made of lipopolysaccharides, LPS, O-polysaccharides, which function as an antigen, and lipid A, which is an endotoxin embedded in the top layer. Gram-negative cell walls will have porins, which are made of protein, to form channels through the membrane. This is an illustration of a gram-negative cell wall. Cell walls and the gram-stain mechanism. As mentioned previously, the structure of the cell wall can be used to sort microorganisms into two specific groups, gram-positive and gram-negative. Crystal violet iodine crystals form inside of a cell during a gram stain. If a cell is gram-positive, alcohol can dehydrate the peptidoglycan in the cell wall, but the crystal violet iodine crystals do not leave. If a cell is gram-negative, alcohol dissolves the outer membrane and leaves holes in the peptidoglycan. During the decolorization step of gram staining, the crystal violet and iodine will wash out, leaving cells that are colorless. In the last step of the gram staining procedure, safranin is added to the cells, and the cells will stain pink. This is an illustration of gram-positive and gram-negative cells. Gram-positive cell walls contain two rings in the basal body of their flagella, produce exotoxins, have high susceptibility to penicillin, and can be disrupted by lysozyme. Gram-negative cell walls have four rings in basal body of the filament, produce endo and exotoxins, and have a low susceptibility to penicillin. Atypical cell walls. Some microorganisms possess what we might refer to as an atypical cell wall, such as Mycobacterium, which possesses an acid-fast cell wall. An acid-fast cell wall is similar to a gram-positive cell wall. However, they possess a waxy lipid mycolic acid bound to their peptidoglycan. They must be stained with carbofuscin. This is a picture illustrating Mycobacterium tuberculosis in an acid-fast stain. Other organisms, such as mycoplasmas and the archaea, also have atypical cell walls. Mycoplasms lack cell walls completely. Sterols in the plasma membrane give it its support against osmotic environments. Archaea can be wall-less or have walls of pseudomerin, which lack an acetylglucosamine and D amino acids. Damage to the cell wall nearly always spells the end for prokaryotic cells. Lysozymes can hydrolyze the bonds in the peptidoglycan, causing the cell wall to break down. Penicillin can inhibit peptide bridges in the peptidoglycan. In either of these cases, without a cell wall, the prokaryote can succumb to the osmotic environment in which it is present and either explode or shrivel to the point where it dies. Protoplast is a wall-less gram-positive cell. A spheroplast is a wall-less gram-negative cell. L forms are wall-less cells that swell into irregular shapes. Structures internal to the cell wall. The plasma membrane. The plasma membrane, also known as the cytoplasmic membrane, is made up of a phospholipid bilayer that encloses the cytoplasm, peripheral proteins on the membrane surface, and integral and transmembranal proteins which penetrate the membrane. This is an illustration of where the plasma membrane can be found. This is an illustration of the architecture of the basic plasma membrane. In it, notice the phospholipids and aforementioned proteins. With regard to the base structure of the cell membrane, it was once thought to be a static, hard structure, not all that dissimilar to an eggshell. Upon further review, it has now been characterized through what we refer to as the fluid mosaic model. The membrane is a viscous fluid structure with a viscosity near olive oil. Proteins can move freely for various functions in any direction in the membrane. Phospholipids can rotate and move laterally. The membrane itself is also self-sealing. The plasma membrane is selectively permeable, which means that it will allow the passage of some molecules but not others. It contains enzymes for ATP production. The plasma membrane can, in some cases, also possess photosynthetic pigments on foldings called chromatophores for the process of photosynthesis. Damage to the membrane by alcohols, quaternary ammonia, detergents, and polymyxin antibiotics causes leakage of the cell contents and the death of the cell. Movement of materials across membranes. When moving materials across the cell membrane, you might employ both passive or active processes. Passive processes allow substances to move from high concentration to low concentration without the expenditure of energy. Active processes allow substances to move quite literally in the opposite direction from low concentration to high concentration, and energy is required. Simple diffusion is an example of a passive process that moves material through a cell membrane from an area of high concentration to low concentration. Simple diffusion will continue until molecules reach equilibrium. Once equilibrium is reached, molecules will continue to move, but they will move from one side of the membrane to the next proportionally. This is an illustration of passive diffusion. Facilitated diffusion is a form of passive diffusion where solutes will combine with the transporter protein in the membrane and move from high concentration to low concentration without the expenditure of energy. This is an illustration of facilitated diffusion. Osmosis is also a passive process. Osmosis can be described simply as the diffusion of water water moving from an area of high concentration to an area of low concentration through a lipid bilayer. This is an illustration of osmosis taking advantage of an aquaporin and the spaces between the phospholipids to move from an area of high concentration to an area of low concentration. Osmotic pressure. This is the pressure needed to stop the movement of water across the membrane. This is an illustration of a combination of simple diffusion and osmosis occurring in a beaker. Note that in either case, molecules will want to move from an area of high concentration to an area of low concentration, either into or out of the cellophane sac illustrated in this picture. Osmosis creates osmotic environments, referred to here. Isotonic, hypotonic, and hypertonic solutions occur as a result of osmosis. In an isotonic solution, solutions' concentrations are equal inside and outside of the cell, and water is at equilibrium. In a hypotonic solution, the solute concentration is lower outside than inside the cell, and water moves into the cell. Hypertonic solutions are opposite hypotonic. The solution concentration is higher outside of the cell than inside, and water moves out of the cell. This is an illustration of each of the osmotic environments I've just described. Active transport. Active transport requires a transporter protein and ATP. It also goes against the concentration gradient. Group translocation, which is a form of active transport, requires a transport protein and phosphoenolpyruvic acid, or PEP, which is a substance that is altered as it crosses the membrane. The cytoplasm. The cytoplasm is the substance inside the plasma membrane, 80% water plus proteins, carbohydrates, lipids, and ions. The cytoplasm also has the cytoskeleton within it. The nucleoid. The nucleoid is likened to a nucleus, but not a true nucleus. It is the location of the bacterial chromosome, which is a circular thread of DNA that contains the cell's genetic information. Plasmids may also be present in the nucleoid. These are extrachromosomal genetic elements that carry non-crucial genes, such as antibiotic resistance and the production of toxins. Prokaryotic cells possess ribosomes. Ribosomes are not membrane-bound organelles. They are, instead, a conglomerate of RNA. They are the sites of protein synthesis made of protein and ribosomal RNA. Through the combination of a 50 and 30S subunit rRNA structure, ribosomes are constructed. This is an illustration of the architecture of a prokaryotic ribosome. Prokaryotes have a variety of other inclusions, named here, metachromatic granules or volatin, which creates phosphate reserves for the cell, polysaccharide granules, which are energy reserves, lipid inclusions, sulfur granules, which are also energy reserves, carboxysomes, which house rubisco enzymes for carbon dioxide fixation during photosynthesis, gas vacuoles, which are protein-covered cylinders that maintain buoyancy, and magnetosomes, which contain iron oxide inclusions to destroy hydrogen peroxide, which is a natural byproduct of cellular respiration. Prokaryotes can also produce endospores. Endospores are likened to plant spores and can be used as a form of reproduction. Resting cells is probably the best way to describe an endospore. Endospores are produced when nutrients are depleted and the cell is at risk of death. Endospores are resistant to desiccation, heat, chemicals, and radiation. Endospores are common in Bacillus and Clostridium species. Sporulation is the formation of an endospore. Germination is how we describe an endospore that is returned to a vegetative or growing state. This is an illustration of an endospore in Bacillus subtilis. Flagella and cilia. Flagella and cilia are projections used for locomotion or moving substances along the cell surface. Flagella are long projections and are often few in number. while cilia are short projections and numerous. This is an illustration of a comparison between cilia and flagella as found in a eukaryotic cell. Both cilia and flagella consist of microtubules made of the protein tubulin. Microtubules are organized as 9 pairs in a ring plus 2 microtubules in the center, sometimes referred to as a 9+2 array. They allow the flagellum to move in a wave-like manner. This is an illustration of the 9+2 orientation in a eukaryotic flagellum. The cell wall and glycocalyx of a eukaryote compared to a prokaryote. The cell wall of a eukaryote is typically only found in plants, algae, and fungi. Animal cells or animal-like cells do not have a cell wall. It is typically made of carbohydrates such as cellulose, which is found in plants, chitin in fungi, glucan and mannan in yeast. Glycocalyx may also have carbohydrates bound to proteins and lipids and is typically found in animal cells. Moving deeper into a eukaryotic cell, we'll look at the plasma membrane. The plasma membrane of a eukaryote as compared to a prokaryote is very similar. It's similar in structure in that it is made up of phospholipid bilayers and integral and peripheral proteins. The main difference between a eukaryote and prokaryotic cell membrane are the sterols, which are complex, and carbohydrates that are used for attachment and cell-to-cell recognition. Both the prokaryote and eukaryotic membrane are selectively permeable and can participate in simple diffusion, facilitated diffusion, osmosis, and active transport. The eukaryotic cell membrane has some other differences in function in that it allows for endocytosis, phagocytosis, and pinocytosis. Endocytosis is a general term to describe the behaviors of phagocytosis and pinocytosis. Phagocytosis is a process where pseudopods extend and engulf particles being drawn into the cell in a vesicle. Pinocytosis occurs when the membrane folds inward to bring in fluid and dissolved substances. The cytoplasm. Again, when compared to a prokaryote, the cytoplasm of a eukaryote bears a number of striking similarities. The cytoplasm is the substance inside the plasma and outside the nucleus. The cytosol is the fluid portion of the cytoplasm. The cytoskeleton is made up of microfilaments and intermediate filaments, which give a eukaryotic cell its shape and support. Cytoplasmic streaming can be used for movement in eukaryotic cells. Eukaryotic cells will also possess ribosomes, just as prokaryotes do. However, their architecture is slightly different as they are made up of large 60S and small 40S subunits. Ribosomes in a eukaryote can be free-floating or membrane-bound as they are attached to the endoplasmic reticulum, forming what we call rough ER. The organelles of a eukaryote. A eukaryote has a true nucleus, which is a double membrane structure encased in a nuclear envelope that contains the cell's DNA. DNA is complex with histone proteins to form chromatin. During mitosis and meiosis, chromatin condenses to form chromosomes. This is an illustration of a eukaryotic nucleus. Eukaryotes also have endoplasmic reticulum in two forms, rough and smooth. The endoplasmic reticulum is contiguous with the double membrane of the nucleus. The rough ER is studded with ribosomes and is a site of protein synthesis, while the smooth ER lacks ribosomes and synthesizes cell membranes, fats, and hormones. This is an illustration of the endoplasmic reticulum. The Golgi Complex. This is a transport organelle which modifies proteins from the endoplasmic reticulum. It transports these modified proteins via secretory vesicles to the plasma membrane in a process we refer to as blebbing. This is an illustration of the Golgi complex, also sometimes referred to as the Golgi body or just plain Golgi. The mitochondria is a double membrane structure which contains inner folds referred to as crista and fluid matrix. It's involved in cellular respiration or ATP production. Lysosomes and vacuoles. Lysosomes are vesicles formed in the Golgi complex which contain digestive enzymes. Vacuoles are cavities in the cell formed from the Golgi complex. They bring food into the cell and provide shape and storage. This is an illustration of the mitochondria. Chloroplasts. Chloroplasts are the location of photosynthesis in eukaryotic cells that possess them. They contain flattened membranes called thylakoids, which contain chlorophyll. This is an illustration of a chloroplast. Eukaryotic cells also possess perioxysomes, which oxidize fatty acids to destroy hydrogen peroxide, and centrosomes, which are networks of protein fibers, and centrioles. Centrosomes form the mitotic spindle, which is critical to cell division. Finally, we'll look at the evolution of eukaryotes. According to scientific theory, life arose as simple organisms 3.5 to 4 billion years ago, with the first eukaryotes evolving 2.5 billion years ago. According to scientific speculation, it is thought that bacterial cells, prokaryotic cells, inhabited the earth first, and as conditions changed, food became scarce, some prokaryotic cells turned to a form of cannibalism, ingesting other prokaryotic cells. So, large bacterial cells theoretically engulfed smaller bacterial cells to develop the first eukaryotic cells. If a cell ingested photosynthetic bacteria, the photosynthetic bacteria became chloroplasts, and those cells followed a plant or algal developmental line. If the cell ingested aerobic bacteria, those bacteria became mitochondria, and the cell then followed an animal developmental line. This is an illustration of what scientists think might have happened about 2.5 billion years ago. And this concludes our coverage of prokaryotes and eukaryotes.